Shalom Maya and Ronen, and to all the students joining us. You ask me how bioinformatics might help determine whether a physician infected a group of his own patients with a hepatitis C virus. To perform such a forensic study, we would compare the RNA sequence of the virus isolated from the doctor with that of isolates from some of the infected patients. So let me tell you about sequence comparison. In this segment, I will first introduce DNA and protein molecules as linear sequences, and then talk about biological sequence comparison in general. In the next segments, we will then describe, with more details, how to compare and align a pair of sequences, how to align multiple sequences, and how to construct a phylogenetic tree that can help us trace the source of infection. Linear sequences are fundamental in biology. DNA is a long double helical molecule that is composed of 4 nucleotides: adenine, cytosine, guanine, and thymine. We can refer to these building blocks as A, C, G, and T. RNA is a single-stranded molecule that is also comprised of adenine, cytosine, and guanine, though thymine is replaced by uracil. So its bases are A, C, G, and U. Therefore, we can treat both DNA and RNA as a linear sequence with an alphabet of 4 letters. Usually, we want to compare genes, which are the DNA sequences that encode for proteins. In some situations, we may wish to compare the sequence of the protein themselves. Proteins, which are molecules with a complex 3-dimensional structure, are also composed of linear sequences, but these are linear sequences of amino acids. There are 20 different types of amino acids, so proteins can be considered as sequences comprised of an alphabet of 20 letters. As we will see, we can basically use the same computational concepts to compare DNA, RNA, or protein sequences. But before we go into the technicalities of sequence comparison, we must explain why we want to compare sequences. Why do we care if 2 proteins, say one from humans and one from yeast, have similar sequences? Well, the answer is simple. Empirically, we know that if 2 genes or 2 proteins have similar sequences, then they probably share evolutionary origin, and they may share similar structure and function. Many studies have shown that the higher the sequence similarity is between 2 sequences, the higher is their structural and functional similarity. We know that during replication of the genetic material, mistakes occur, which we call mutations. These mistakes in the replication process result in either an insertion, in which a nucleotide is added to the sequence, a deletion, in which a nucleotide is lost from the sequence, or substitution, whereby a nucleotide is replaced by another nucleotide. DNA mutations are rare during each replication cycle of a genome, but they accumulate over evolutionary timescales. Thus, when we look at the starting sequence and the sequence we observe following an evolutionary process, the 2 sequences may look quite different. Nevertheless, our methods should be able to determine that the sequences are similar in the sense that a relatively small number of mutations was needed to change one DNA sequence to the other. In fact, the number of mutations needed to convert one sequence to the other may be used as a measure of their similarity. Remember that changes on the DNA level can lead to changes in the resulting protein sequence. Let's look at example 1. Are these 2 sequences similar? Sequence 1, which spells the word peace, is actually a sequence of the 1-letter codes for the amino acid proline glutamic acid, alanine, cysteine, and glutamic acid again. Usually, proteins contain hundreds of amino acids, but here, to illustrate these concepts, we will use very short sequences. Sequence 2 is also PEACE. So these two sequences are not only similar, they are identical. We can therefore say that they are 100% similar. What about example 2? Are these two sequences similar? We have peace versus fight, which represents the one-letter code for the amino acid phenylalanine, isoleucine, glycine, histidine, and threonine. These two sequences are completely different and do not have even a single amino acid in common. So these two sequences must be 0% similar. Those were the easy questions. Now let's look at example 3. What is the similarity of these two sequences? They are neither identical nor totally different. Thus, we need to find a way to quantify the level of similarity between two biological sequences, as we will describe in the next segment. Let's go back to the reasons why we want to know whether and to what extent two biological sequences are similar. First, we can use sequence similarity to infer the properties of novel proteins. For example, if we have a great deal of biological knowledge about a protein in yeast, and we find a human protein with significant similarity to this yeast protein, then we can assume that the human protein has similar structure and possibly similar function to that of the yeast protein. Note that bioinformatics analysis can't prove the functional similarity, but it can give a much better starting point for experimental verification. Furthermore, even if the two sequences are not similar along their entire lengths, they may have a local region of similarity. For example, a similar domain that could lead us to identify similar function, such as DNA binding. Another reason why I want to describe sequence comparison of two genes or two proteins with a fair degree of detail is that this simple algorithm is a fundamental technique in bioinformatics, upon which many other more sophisticated methods are built. Computer scientists have long been interested in characterizing the similarity between sequences and developed algorithms to compare text in order, for example, to improve spell checkers and text editors. Vladimir Levenstein, a Russian mathematician, developed already in 1965, when very few protein sequences were known and DNA sequencing was not even developed, a measure called edit distance, which forms the basis for biological sequence alignment methods. In the next segment, we will describe quantitative methods for the three tasks that we have mentioned: pairwise sequence alignment, multiple sequence alignment, and constructing phylogenetic trees. Based on this understanding, I think it will become clear how bioinformatics can help identify the source of infection, as well as the utility of sequence comparison to address many other biological and clinical applications.